For over a century, the core rule of infection has remained the same. To replicate and spread, a pathogen needs genetic material. Whether it's a virus, a bacteria, or a parasite, every biological threat carries a payload of instructions in the form of DNA or RNA. This genetic payload is also a target. By using heat, chemicals, or medicine to shred those instructions, we can neutralize the threat and stop an infection in its tracks. The prion, however, lacks every one of these features. It contains no DNA, no RNA, and possesses no cellular structure. Under a microscope, it appears to be nothing more than a single inert protein drifting through the body. Despite being a non-living molecule, a prion is highly contagious, and once an infection begins, the fatality rate is 100%. This forces us to answer a specific question. How does a molecule with no genetic instructions manage to systematically and unstoppably destroy a human brain? Prions aren't invading organisms. They are actually misfolded versions of proteins we already have. These normal prion proteins are found naturally throughout the human body, specifically concentrated within the healthy cells of our nervous system. An infectious prion occurs when one of these normal proteins suffers a structural error. It misfolds, and instead of remaining flexible and functional, it locks into a rigid, jagged shape. This misfolded protein then acts as a template. When it collides with a healthy, normally folded protein, it physically forces that neighbor to snap into the same jagged, rigid configuration. This creates an exponential chain reaction. The misfolded shapes multiply through the tissue, spreading at a rate that mimics a viral infection, but without any actual cellular reproduction. This mechanism turns our most basic biological building blocks into the agents of our own decay, as a single geometric error begins to consume the surrounding tissue. As these misfolded proteins clump together into dense plaques, they become toxic to neurons. Cells begin to die off, leaving behind microscopic, expanding holes in the brain tissue. This physical degradation is known as spongiform encephalopathy. As the brain literally takes on the texture of a sponge, the patient experiences rapid onset dementia, sudden involuntary movements, and the loss of basic motor control. Once these clinical signs appear, the decline is irreversible. The typical duration from the onset of symptoms to death is only 4 to 5 months. We have seen the impact of this process on a massive scale. Bovine spongiform encephalopathy, or mad cow disease, spread through cattle populations in the 1980s after they were fed contaminated agricultural products. When humans consumed meat from those infected cattle, it triggered a variation called variant Creutzfeldt-Jakob disease. The bovine prions acted as templates for human proteins, beginning the same fatal chain reaction in the human brain. The disease has even been found spreading through ritualistic cannibalism in Papua New Guinea, a variation called kuru, proving that these rogue proteins can find multiple routes into a host. Regardless of how the first misfolded protein appears, whether it's inherited, whether it's consumed, or occurs sporadically, the result is a brain physically hollowed out by a microscopic shift in geometry. To protect patients, hospitals use extreme sterilization protocols. Surgical tools are placed in autoclaves and subjected to high-pressure steam, intense heat, and corrosive chemical baths to destroy any biological life. Prions easily survive these standard methods. They remain infectious even after being boiled, radiated, or subjected to the intense heat of a medical autoclave. This resilience exists because prions have no cell walls to rupture and no DNA to shred. Our most advanced sterilization tools are designed to kill living things, but a misfolded protein is already inanimate. This invulnerability turns ordinary medical equipment into a potential vector for the disease, requiring specialized, aggressive decontamination methods just to break the protein structure apart. Prions demonstrate the most fundamental rule of molecular biology, that in the microscopic world, a molecule's entire function is determined by its three-dimensional shape. A microscopic shift in how a single strand of amino acids is folded can lock a common building block into a static, impenetrable form, one that the body's natural waste-clearing systems cannot break down. Despite our understanding of how this chain reaction works, there is currently no medical treatment that can stop or even slow the progression of the misfolding once it begins. The result is a pathogen that is effectively indestructible. It is a fatal miscalculation occurring deep within our own biology.